All right, everybody. It is my great pleasure to introduce Dr. Sujal Phadke to the CSU virtual seminar series. And Sujal completed her undergraduate studies in India before coming to the US and obtaining a PhD in ecology and evolutionary biology at the University of Houston. Please correct me if any of this is wrong.
Perfect.
Uh, she completed two postdocs, one at Duke and the other at the University of Michigan, before moving to a career in nonprofit research, which I'd love to hear about that. And then was a staff scientist at JCVI, the J. Craig Venter Institute in San Diego, which she somehow survived. No, I'm sure. I've heard funny things about it, though. I really have. And at Rady's Children's before joining the faculty at our Cal State CSU San Marcos, where she works on microbial evolution, genomics, and bioinformatics, and of some of my favorite and most underrepresented, I think, in the microbiome world, creatures, which are the microbial eukaryotes that are much more diverse and far more interesting than anyone gives credit for. So I am really excited to hear about this because whenever I hear about microbial eukaryotes, I'm like, oh, people should study this more because they are massive numbers of them, and a lot of our worst parasites are microbial
Yeah,
eukaryotes.
yeah,
And they're so weird. They're just weird. But most of, as I understand, most of eukaryotic diversity is actually single cellular.
that's right.
Yeah. So, and we just only think about the charismatic megafauna. So, anyway, that's my little pitch to say how cool this is. And please go ahead and take it away.
Thanks, God. I appreciate the enthusiasm about these organisms because, like you said, very few people really think about it or get to know about them. And there are so many of them. It's almost impossible to study everything that's out there. But hopefully, I will talk about two interesting cases today. So I've been working on microbial eukaryotes, different kinds, since PhD. And I'm mostly interested in their survival strategies. So that's essentially what I'm going to talk about today. Um, Overall, I love to study differentiation in these species. And when we talk about differentiation, we don't necessarily talk about microbial eukaryotes or unicellular species, single-cell organisms. So I would like to kind of take an approach where I first introduce everybody to what microbial eukaryotes even are, because it seems like everybody uses this term a little differently for good reasons. And then I will talk a little bit about what differentiation really means in these type of organisms. And then most of the time I'm going to spend talking about the two ongoing projects, and that ongoing is a very important word there, which means it's work in progress, much to be done, but we have learned some cool things about these species already. So in the first project, we are looking at a few species of marine haptophytes, and we are trying to figure out if they're having sex. And so some bioinformatic tools there help us kind of figure out answer to this question. And in the second project, I'm going to talk about ciliates, which is another group of supremely diverse unicellular eukaryotes. But these are the organisms I worked on in my PhD, and so I've kind of continued working on them or exploring because just they're so super cool. Before I go any further, though, I want to acknowledge and really emphasize the efforts of three people here, mainly Aihotu and Asher. So Aihotu is a graduate student here at CSU San Marcos, and Asher is an undergraduate student. And the work that I'm going to be talking about today is basically theirs, and I'm just talking about it. So it's something that I have learned a lot about as well. For example, especially in marine haptophytes, I'm a kind of a newbie to haptophytes life cycles, and Asher, he's amazing. It's like ideal student, great expert at microscopy, amazing. Knowledge of these species, and I have learned a lot from him. And Aihotu has just been super phenomenal in all of the bioinformatics analysis. So I'm going to be talking about their work. And then I'm also very happy to say that because I continue working on ciliates, I get to also collaborate or continue collaborating with my PhD advisor, Dr. Becky Zufall. And so after a long time, we are collaborating again on this project. So I want to thank her as well quite a bit. So let me introduce microbial eukaryotes. There is not a single definition per se. Really, any microscopic cell, that is eukaryotic cell, that lives as a single cell usually is a microbial eukaryote. But that's not really the only kind of definition there is out there. Most of these are unicellular protists. But there are some fungi that are microscopic, that are single celled, or at least have predominant life stages that are single cell, and they also can be considered under this big umbrella term of microbial eukaryotes. So this is how I define it to myself. A bunch of unicellular protists in different clades in the tree of life. So here you see the tree of life, and I have kind of marked all of the clades that include some of these organisms. And like Scott was saying, these constitute the most diversity of eukaryotes. So you see protists in these first two clades. The first clade is like Giardia and relatives. The second clade here, alveolates and straminophiles. That's where ciliates are. So that's what the second project is going to focus on. That is also where many of the parasites are. So malarial parasite, for example, is a cousin to ciliates. And there are other parasites in this group as well. The amoeba and slime molds, they're also considered under protist. So protist is really not a monophyletic group per se. Algae, different kinds of algae are also protists. And so really anything that is not a plant, that is not a fungus, and that is not a metazoan is a protist. But the microbial eukaryotic term would include protists as well as some fungi. And I actually worked on fungi in my postdoctoral years, and I continue working on them. But that project I'm not going to talk about today in the interest of time. So really the only two projects I will focus on today is first, I'll talk about marine haptopods. The reason there are two arrows that are here is because their phylogeny is not completely clear. It's really hard to study them. And so there are some disputes about whether they should be included in this clade versus that clade. And then ciliates would be the second project that I would be talking about. So thinking about differentiation, we don't really use this word differentiation much when we talk about eukaryotic microbes. Usually it is used in the context of multicellular species like us, where you have some very specific distinction between, you find two types of cells, or at least two types of cells in the body. There is something called as germline. For us, those are usually eggs and sperm. And then there are many types of somatic cells. And so most multicellular organisms have this distinction very clear. You also have this whole developmental pathways going from stem cells or embryonic cells that differentiate into different types of cells. And a lot of times that is terminal differentiation. So this de-differentiation pathway is blocked many times as well. And so when it comes to multicellular organisms, differentiation is a little different than when we talk about single-celled organisms. In multicellular organisms, you have both reversible and irreversible stages that are differentiated. So you can differentiate into that stage and go back to the original, or you're terminally differentiated. In single-cell organisms, so in most microbial eukaryotes, most of the times differentiation is reversible. So it's a really nice summary from this paper by Wagner. You have some proliferative cell state. Usually it's an asexual reproducing cell. And then it experiences some stress, maybe starvation, maybe one particular nutrient is low. Maybe it's just an environmental shock. Whatever the stressor might be, usually that induces some sort of differentiation, and it enters this quiescent cell state, which usually is a state where it is spending a lot of time surviving that stressful condition. And when benign environment returns, it de-differentiates and starts reproducing again asexually. So most of the differentiation is reversible. In microbial eukaryotes, it is also an adaptive response to different stressors. And many times for many species, we don't know the environmental triggers that would cause this differentiation. But sometimes, especially in the case of parasites, and I have an example here. So this is on the right, it's a life cycle of Nigularia, which when I first learned about it, the first time I learned, I think I was an undergraduate or maybe early stage of graduate. school, and it's the brain-eating amoeba. And so it shows this remarkable differentiation most of the times in natural environment, in aquatic type of habitats. It exists in this cyst form, which is really a resistant dormant form. It has a very thick wall covering. And then at some point, it differentiates into this amoeba form. It's also called trophozoite. Um, but it's a typical amoeba, the textbook description of amoeba. And if you look at these two forms under the microscope, you wouldn't know that they're really the same species. They look so different. This amoeba can reproduce, on its own, but at some point it can also turn into a third form, and that's the flagellate form. So it has a very distinct morphology than the first two forms. The cell shape is different, the size is different. There are two clear flagella. And then this is the stage that we, as humans, are at risk of getting infected with during any recreational water-related activities. Once it enters the body, if it survives, it can turn back into the amoeba form and penetrate even our brain. So it actually migrates as amoeba into our brain and literally can eat our brain from inside. So it's a pretty nasty parasite. But what's cool about it is there are so distinct three different forms in this species. And this is by no means an exception. You see this in many microbial eukaryotes. Malaria does something similar. Giardia does something similar. Even non-parasitic microbial eukaryotes do this. Morphologically distinct forms in distinct environments. And that really got me hooked onto these species. Like, how are they doing this? It's a single cell. It's completely transforming itself into another form. And I'll talk more about this when I talk about the first project right after this. But a lot of times these forms look so different that people have, mistaken them for completely different species. And only after the advent of genomics, we got to know that, wait a second, these two forms that, look nothing like each other are in fact two different life stages of the same species. So I think it's really remarkable for a unicellular organism to be able to do this in response to certain environment. And we know very little about what triggers this. So I'm interested in those kinds of questions with different microbial eukaryotes. So the first project I'm going to really talk about is that tries to ask this question about a particular group of microbial eukaryotes, and those are called haptophytes. And like I said, I'm still a newbie in haptophytes. I'm still learning about them. When you are doing bioinformatics, you get to learn about many different organisms, and you get to actually work with many different organisms, which is the best part, I think, of being able to have bioinformatics tool set with you. But in this project, what we are trying to do is try to figure out whether these specific groups of species are having sex. And before I get into why we are interested in that and what does bioinformatics have to do with it, let me introduce you to who these organisms are. You probably have encountered them, especially in San Diego. You have encountered them around you. These happen to be some of the most abundant marine phytoplanktons. They're ecologically super important. They're actually also used quite a bit in aquaculture industry as feeds for oyster and some other marine animals. They are also a very diverse group of organisms, but there is one unifying feature that they have. So they have this specific structure called haptonema. It's not really a flagellum, but it's very much alike. So you kind of see that here in this green photograph. None of these pictures are mine, by the way, but they're very beautiful and very representative. So this H thread here, that's the haptonema, and we don't really know the function of it, but sometimes we know that it is used to catch prey. And then it also, many marine haptophytes also have these flagella, and I'll talk about them in a little bit. So haptonema is a unique type of flagellum that is different from the typical flagella we talk about. And that's really their pretty much only unifying structure of these species. Other than that, many of them, not all, but many of them, they form these beautiful calciferous shells. And so there is this electron micrograph of different species that shows these different types of shells, and really it's like one shell and multiples of that kind of covering a cell, and each of these shell plate, as I call them, is called a coccolith. So there are multiple coccoliths kind of covering an entire cell, and depending upon what species it is, you get these different shapes of coccoliths. I'm also going to talk about coccoliths in just a bit when talking about differentiation in these species, but when I said you have encountered these around here, especially in San Diego, is because many species of haptophytes, or at least some very characteristic species, cause what those toxic algal blooms, those beautiful blue tides that are not really good for the environment, but especially in the Southern Ocean. That you see these belts where it's called great calcite belt. That's because of their calcifying or calciferous shells, and they're abundant in there. And every once in a while, seasonally, you get to see those blue tides thanks to some of these coccolith producing species. There's a really great movie about how they produce coccoliths. Unfortunately, it's not playing within the PowerPoint, and I'm just going to leave it there. If you honestly just Google on YouTube as coccolith forming species or haptophyte. This is a video by Dr. Allison Taylor, and you would be able to find it. I highly recommend watching that movie. It's like watching a cell do something supremely personal that we wouldn't get to see otherwise. It's a wonderful video. And just to kind of give you an idea, what you see in this snapshot of the video, it's the cell of one of the species of coccolith forming haptophytes. And then this part right here, that's actually one of the shell, the coccolith that it has just produced. And actually inside the cell, you see the other one being produced. And in the video, if you watch it, you will see that that one pops up and gets outside and attaches to the cell. Then the third one pops out, then the fourth one pops out, and one by one, this cell is covered in coccolith. So it's a pretty cool process how they create these giant calciferous shells. So what are the specific things I'm interested in when it comes to these haptophyte species? So they seem to have a life cycle that has two distinct phases, and that got me interested in it because those two distinct phases, as you might have guessed, they look starkly different. These are some of the pictures that Asher took here in the lab and also at SDSU electron microscopy facility. What you see here, so this the red circle here that reflects what we call a diploid vegetative growth, and it has one specific type of coccoliths. You see the electron micrograph here and the light microscopy image down here, where you can see the coccoliths kind of covering the cell. This is the non-motile stage. So if you look at them under the microscope, they just kind of stay there. But at some point, with some environmental triggers that we unfortunately don't know what they are, this diploid stage gets converted into this other life cycle stage, which is the haploid. That can also grow vegetatively, but it looks very different. So this haploid stage, as you can see here in the picture, and also in the cartoon, it has much fewer coccoliths. There are some, but you can't really find them because there are a lot fewer than the diploid stage. So the coccolith distribution changes going from diploid stage to the haploid stage. But also the cell becomes smaller in size, which is something you can't see here. But what you can see that it also differentiates into this flagellated form. And so this stage, the haploid stage, is motile. So this really got me into thinking, it's like, how is this happening? If somebody had showed me these two cells without knowing that they are of the same species, I would have probably, not thought about these two being the exact same, or two life stages of the exact same species. We think, based on some prior knowledge that we have, not necessarily of the haptophytes, that this transition occurs because at some point this diploid cell is undergoing meiosis. And as we know, when you undergo meiosis, you reduce ploidy, you halve the ploidy really. So you go from diploid to haploid. But during this process, you also differentiate morphologically in many different traits. And so it's a hypothesis that it is the meiosis that is causing this. We don't know for sure. So if we want to know for sure that this is really meiosis, how do we study it? And that's where I started thinking about, let's see if we can answer this question. Is it really meiosis that is causing this phase transition from diploid stage like non-motile kind of to motile haploid stage. And the question is, why wouldn't it, right? It seems likely going from diploid to haploid, meiosis might be the right answer there. And it's true, there are some other species, not necessarily of haptophytes, but closely related species that have been shown to undergo meiosis and do exactly the same thing or very similar thing. But we also know that there are other ways to reduce ploidy. And so this is a really nice summary from this paper by Anja Forche, which shows three kind of ways to reduce ploidy, where different microbial eukaryotes reduce ploidy by undergoing a process that is non-meiotic. So here, the first thing that is sexual, that is canonical meiosis. You have mating, you have some meiotic products that mate, and then you form the diploid bag, and that diploid can undergo meiosis. So this is what we think might be happening. We don't know for sure. That's what we want to know. But we also need to think about what are the alternative ways of this happening. And so the two alternative ways are this parasexual cycle, as we call it, or asexually, really. And what happens in either of these cases, there is no meiosis. There is just chromosomal non-disjunction. Sometimes it is chromosomal loss. A lot of times it is random. And so at the end of this process, through multiple chromosomal losses, you essentially reduce the ploidy. There is no meiosis per se. There is nothing about sexual about these B and C options here. So, and many microbial eukaryotes actually do it. So there is a possibility that the haptophyte species that we are studying, they might be doing that. And so it would be really nice to know is it really meiosis? Are they having sex? Because, so meiosis is defined as sex, because sadly, most of the haptophytes, we can't really catch them in action in the lab. We can't really grow them enough in the lab. We don't know the environmental triggers that will trigger this phase transition. And so we have never really caught them into action. So the question is there any other way? If we cannot make direct observations, is there a way that we can figure out, are they having sex? Meaning, are they having, undergoing meiosis? Are they even capable of undergoing meiosis? And so that's where bioinformatics really helps, because if you have organisms that you can't really grow in the lab, because you haven't figured out how to grow them in the lab, they're not really amenable to laboratory cultivation. How do you study life cycle of those species? Well, you can do quite a bit using simply in silico bioinformatics genome kind of mining kind of analysis. And that's exactly what we are doing here. Our approach has been to take a bunch of genomes of haptophytes. We will have about 35 of them. We don't currently. But once we have these genomes, what we are going to do is we are going to inventory them for the presence of genes that are important for meiosis. Because the hypothesis there is that if you have genes that are important for meiosis, then you are likely capable of doing meiosis. And that's probably the predominant process that is driving this phase transition. So what we have done so far is we start by obtaining sequences about around 60, I think the exact number is 57, but around 60 genes that we know are important for meiosis in different species. So there are genes that we know in diatoms, which happen to be closely related group. We know that those genes are important for meiosis in diatoms. And we, of course, know genes that are important for meiosis in yeast and plants. So we gathered sequences of all of those genes. And then we use those sequences to query our genomes to simply ask, are homologs of these genes present in haptophyte genomes? And the tools are really simple. We start with BLAST, protein BLAST. And if we find a homolog with a really good match, then that's great. We use reciprocal BLAST to kind of verify or confirm the homology, but also for genes that are really divergent, you can also use Hummer approach and see if you can find homologs of those genes in haptophyte genomes. So that's essentially what we have been doing. At the end of this process, we get essentially a giant table, which is what we call inventory. You have like a list of genes, all of the 60 genes, and then you have a list of genomes, and we really just say plus if the homolog is present, and minus if the homolog is absent. Of course, this is not the end of the process. You then have to take every gene and kind of do phylogenetic analysis, basically build phylogeny to infer whether these are orthologs, paralogs, to kind of confirm. I'll show you an example of how that looks like. But this is the whole process as far as bioinformatics is concerned. Of course, you should never stop at bioinformatics analysis. There has to be some kind of functional evidence that if you find meiotic toolkit genes, and if you're claiming that meiosis is indeed happening or these species are capable of meiosis, then it would be good to actually, show that these genes are functional. So we are also working on gathering functional evidence, albeit that work has been really slow because what it involves is that you have to actually kind of figure out the way to grow the diploid stage in the lab, the haploid stage in the lab, and then you take a transcriptome of the diploid stage, take a transcriptome of haploid stage, and see whether these genes are showing stage-specific expression, because meiotic genes do tend to show stage-specific expression. So that is the part I'm not going to talk about today because we don't have those data yet. But I will show you a little bit about what we found when we did inventory of some of these species that we already have genomes for. So we have genomes for about 5 species so far. The others are being sequenced. Some of them are done, but they're not annotated yet, so on and so forth. So what I'm going to be showing you is inventory of these 5 genes. So the 3 species are in the Pavlova genus, which are up here. 2 species from the Emantonia genus, which are here, and then we have a genome of the outgroup. So we have also inventoried that. So that outgroup is Glossomastix. It's a closely related straminophile. But we will be getting in future genomes of all of these species here. And we know quite a bit about these species. We know where they live. Some are freshwater, not all are marine. Some are polar, so they live in different habitats. Some of them are calcifying, some of them are not calcifying, so on and so forth. So we can make a lot of correlates between who is having sex, or just the calcifying one having sex. We can do a lot of analysis in terms of correlations. But so far, we are looking at these 5 species. None of them are, calcifying. Unfortunately, we also do not have both haploid and diploid stages in the lab for these species. So the genomes that we inventoried are exclusively coming from the diploid stage right now, at least as far as these 5 species are concerned. So once we go through all of the homology searches, the reciprocal blast, this is the giant table I was talking about that we end up with. And so it's a lot to take in, and you don't have to memorize any of this, but I'll walk you through, or at least highlight some of the important aspects. So this first column is essentially different genes that are included in meiosis toolkit. So you see some genes are important for DNA replication and maintenance, double-strand break repair, crossover regulations, so on and so forth. So those are groups of genes for those individual processes that are involved or related to meiosis. And then each column here is a distinct species. So somebody did a very similar study for diatoms, 6 diatom species, and I have kind of included their data here that was published in 2015 as well, just for comparison. And then the haptophyte data is ours, and also the green one is the Glossomastix, which is the outgroup that we are using. And so here are the 2 Emantonia species, IM1 and IM2. Here are the 3 Pavlova species, PV1, 2, and 3, and GL is our outgroup. So one thing that you can take from here is there are a lot of genes that are present in a lot of species, but there are also a lot of genes that we couldn't find in both our haptophytes, and also Patil et al. Couldn't find them in diatoms as well. And so there is a lot of lineage-specific variation in these genes. Some genes are absent from some species, but present in other species, so on and so forth. But there is one pattern that is really interesting, and I have highlighted those sets of genes here in red. So these XRS2 and this group of genes here, those genes that are highlighted in red, if you look through the entire sort of set of species, you realize that they are absent in all of them. They're absent in diatoms, they're absent in our haptophytes, they're also absent in our outgroup. And so we started wondering, what are those genes? And turns out those are the genes that are coding for proteins that are involved in synaptonemal complex. And if you don't know what synaptonemal complex is, that's totally fine. I'll talk about it a little bit. But the take-home message here is that there are a lot of meiotic genes that are present in our haptophyte genomes. And so likely the haptophytes that we have, at least the ones that we have inventoried so far, they are very much capable of meiosis. So indeed, it could be meiosis. But meiosis itself can occur in many different ways, at least two major ways. And so one is with synaptonemal complex. That's the kind of meiosis you find in, for example, us or metazoa or these model species. But there is another pathway of meiosis, and that is without the synaptonemal complex. And it has been frequently seen that many microbial eukaryotes actually adopt that pathway. And so it's interesting to see that in our data set as well. Of course, these are just 5 species that we have analyzed so far. We have 30 more coming in, but it should be interesting to see that. Before I get into the synaptonemal complex, I do want to just show you an example of once you get that inventory, what's the next step? Why do you need to build phylogenies in order to kind of infer paralogy or orthology? So here's just an example. We are building phylogenies of each of those 57 genes. But this is a phylogeny of BRCA1 and BRCA2. These are kind of accessory proteins that are involved in, some processes in meiosis. And so what we did was we gathered sequences from Arabidopsis, from human, from mouse for both of these genes. So here is a cluster that is in pink, that is for BRCA2, and here is a cluster in purple that is for BRCA1. And you see that our Pavlova BRCA1. So BRCA1 was only found in Pavlova. We didn't find it in Hemangtonia, but it clusters with other BRCA1s and BRCA2, which was found in diatoms by Patil et al. And we also found it in Hemangtonia, but we didn't find it in Pavlova. So those BRCA2 also cluster with other BRCA2. So this is the kind of analysis that we are doing once we have our inventory to ensure that these are really the homologs that we think they might be. Okay, so let me talk quickly a little bit about what synaptonemal complex is. Like I said, it is involved in one of the pathways of meiosis. So this is a whole pathway. Don't worry, I'm not going to go over every single detail, but suffices to say that you start the meiotic pathway here. You have a pair of homologous chromosomes that the non-sister chromatids then engage in crossover, so in recombination. And these recombination events, these crossover bridges, are resolved by these synaptonemal complexes. So I have zoomed in a synaptonemal complex here. It's really a bunch of proteins. These are all of the genes that we also inventoried. And these bunch of proteins essentially resolve crossovers to finally get you this configuration, the recombinant configuration here. That's the canonical class 1 crossover pathway of meiosis. That's what happens in most model species. Turns out our microbial eukaryotes, both the ones that we studied, but also those that other people have studied, they lack that synaptonemal complex. And so there is alternate pathway to resolve these crossovers, and that is called class 2 crossover pathway. And it's not a predominant pathway in us or in model species, but seems like it is probably the predominant pathway in microbial eukaryotes. And so we were interested in seeing who else doesn't have this synaptonemal complex? And turns out many protists actually lack this synaptonemal complex. So earlier studies show that ciliates, which I will be talking about in just a minute, they lack the synaptonemal complex. Many species of diatoms, our haptophytes, and even some fungi. So many microbial eukaryotes seem to have figured out how to do crossover regulation even without needing synaptonemal complex. So that generates a lot of questions about the evolution of synaptonemal complex, the necessity and the role of synaptonemal complex. But I'm not going to go there right now. I'm just going to summarize this part of the talk. Basically, looks like at least these 5 species are capable of meiosis. And so if we ever find the haploid stage, it would be, probably because of meiosis. Of course, this is not a confirmatory stage. We need to gather functional evidence and get transcriptomes to see whether these genes that we find are actually functional. Are they being expressed in specific stages? The synaptonemal complex was an interesting finding, but it kind of was consistent with the other studies as well. What I liked the most is that we wouldn't have been able to study or even explore this question if there were no bioinformatic approaches to do that, because we have tried, and Asher can tell you he has tried so much trying to transition some of these species from diploid to haploid stage. In the lab, we tried, different triggers, reduce the light, increase the temperature. Unfortunately, we could not find the triggers so far. That work continues, but at least we can learn it using bioinformatics tools a little bit so that we can keep going. So that was the first project. We still have about 30 genomes coming in. We need to annotate them first. So there is a lot of related bioinformatics stuff that needs to happen before we can even do the inventory. But that's where I'm going to end that, talking about that project. And I want to talk now about the second project. So the second project is not on haptophytes. It's on ciliates. Like I said, ciliates are this extremely diverse group of species. They're monophyletic, and their unifying feature is that every single species has cilia. So that's really their unifying feature. But other than that, they're pretty diverse. Maybe the other unifying feature also is that, well, most of them are unicellular, but also they have this really cool system. They have two nuclei inside the single cell. And one acts more like a germline nucleus, is transcriptionally silent. Ciliatologists call it mic for micronucleus because it's small. But it's really the germline nucleus because it is the one that passes to the next generation during sexual reproduction. But they also have this other nucleus, which we call MAC because it's macronucleus. It's big, and that's the one that is transcriptionally active. So in day-to-day life, the cell uses whatever genome that is present in this MAC. The MAC genome, the somatic genome, does derive from this mic genome from the germline. So there is this great germline soma distinction within a single cell, and you find that in every single species of ciliates, and that's a really good way to separate germline and soma within a unicellular species. But there are other cool things about these ciliates. They have somehow figured out how to use all 64 codons to code for amino acids. They, of course, use some of them as stop codons as well, but those stop codons are also going to be used to code for amino acids, which I think is pretty cool. Some of the species have figured out how to make gene-sized chromosomes, so they actually rearrange and scramble their genome and process their genome to kind of make gene-sized chromosomes, so every chromosome is a gene. They also know how to amplify it up to a certain point. They also have these multiple sexes. Anyway, I can talk about ciliates and why they're cool for any length of time, but they do have a lot of, I feel like I say they live biology by exception, or they do biology by exception. Every textbook definition of anything, ciliates have kind of violated it, and, they have figured out how to live life without it too. So I keep working on them also because they show a lot of different responses to environmental conditions. So this differentiation is also seen in these groups of species. So most of the times they live as vegetatively growing cells. But when you starve them, especially, some species can turn into parasitic forms that look very different. They get this giant nucleus, and these parasitic forms can infect fishes and other aquatic animals. Some, increase in size up to even 5 times and start cannibalizing in the culture. Some just, turn to sexual reproduction. And some actually become dormant forms, and there are 2 kinds of dormant forms. So they're what we call resting cysts, and there are some reproductive cysts. In either of these cases, the cilia are completely lost, other than the cannibalistic form and the sexually reproducing form, which are very similar to the vegetative form. In all the other forms, the cilia are lost. The cell protective covering changes, and so these are very distinct morphs that these ciliates can get into upon certain triggers, starvation being one of them. So this is the process that I wanted to study. And so many ciliates are actually amenable to laboratory cultivation, but there are many that are not either. And so those who are amenable, we have a lot of information about what kind of starvation responses do we see. So one species may show multiple different starvation responses. Sometimes it can turn cannibalistic. Sometimes it can go dormant. We don't know exactly how it chooses, but there are some species that, just adopt one strategy. So they turn parasitic, and then that way they can explore other niches and stay alive or survive. Or sometimes they just turn cannibalistic. So It really depends on the species, what responses do you see. But we decided to ask the question, which of these different starvation responses really seems ancestral in ciliates? Where did it all begin? Because many of these starvation responses, so going from vegetative to each of these survival strategies, there are many overlapping things between these survival strategies. There is CAMP, CAMP, essentially based pathway, protein kinase based pathway. They're a very similar set of genes that are important for each of these survival strategies. And so we wanted to know, well, which one is really ancestral? So we use this character state mapping kind of approach, which really means that you have a phylogeny of these species. And along with that, you have a table, and you have marked down in the table who does what. This one is cannibalistic, this one is parasitic, this one turns to sexual reproduction, so on and so forth. And then you see how those characters are distributed across the phylogeny, and then reconstruct the ancestral trait given the distribution that you currently have in this species. And so I will talk about the ciliate one in just a minute, but I kind of just wanted to give you an example of how it looks like. So this phylogeny is from this paper that is linked here, but they were trying to see how many times bioluminescence has evolved in a specific group of fishes. And they used ancestral character state mapping. They built a phylogeny and they mapped on that phylogeny where do you find bioluminescence and where you don't find bioluminescence. And I think they found out that something ridiculous, like it has evolved 27 times or something like that independently. That tells you some significance of having bioluminescence in these kind of organisms. So there are multiple questions about evolution of a certain trait that you can answer by doing this character state reconstruction kind of approach. And so the approach is really simple. You have a data matrix in which something that looks like here, you have the species, you have each character marked, whether you find it in species or not, a character being any trait. So for example, are they encysting, meaning are they going dormant? Are they turning parasitic? So on and so forth. And then you construct a phylogeny, and on that phylogeny, you map those characters. So there are various approaches to map the characters. Different approaches have different advantages. The one that I'm going to show you is based on maximum parsimony, although we are working on getting maximum likelihood and Bayesian worked out as well. But this is essentially going to give you a tree that looks like this, and then you can say, well, which one was ancestral and which one wasn't. You can also correlate between the different characters. So this is the first kind of tree that we built. This is a tree of pretty much all the ciliate species that we know of. What is marked on this tree, or the character that is, or the trait that is marked on this tree, is how many layers do you have in that cyst wall. So all of these species produce some type of dormant cyst. And if you look at the ultrastructure of that cyst, some species have 4 layers of the cyst wall, some have 3, some have 2, some have 1. And so I have a little cartoon here that shows the cyst wall, depending upon how many layers you have. And then inside that, or enclosed within that wall, are all the good stuff, the nucleus, the mitochondria, the vacuole, so on and so forth. And this form can stay dormant pretty much forever. 10 years ago, I went on a trip while moving between my postdoc positions, collecting soil samples. And to this day, I can get those soil samples, put them into water, and whichever ciliates are there that in the encysted form, they just come out alive, and I get to see them under the microscope. So it's a pretty resistant, dormant kind of structure that can stay on forever. And so ciliates can really survive in this form for a long, long time. And so what this tree shows is that overall across the phylogeny, if you plot the cyst wall layers, it seems like cyst wall with 3 layers is really inferred to be ancestral. Here, there is this giant clade that has 4 walls, so they have gained one kind of layer, but there are then some species that have lost a layer. So going from 3, which is the ancestral state, to 2 layers in some species that you see in green, or even just single layer that you see in red. And so these each layer has a different kind of chemical structure, which means there is corresponding secretion machinery and biosynthetic pathway that we get to see how it has diversified as well. And so there are some lineage-specific gains and losses of those secretory machinery that we can unearth by doing these kind of analysis. We also did another kind of analysis, not across the ciliates, but within one particular group of ciliates. So that's Tetrahymena, and that's the genus that you might know came really to light when two actually Nobel Prize winning discoveries were made using these organisms, so ribozymes and telomerase. They were both made using Tetrahymena. And there are other cool things about Tetrahymena. We also know a lot about Tetrahymena in the lab. And so this is a tree of all species of Tetrahymena that we have known. And it shows which species produces resting cyst, which produce no cyst, and which species produces reproductive cyst. So remember, resting and reproductive are the two type of dormant stages. The reproductive cyst really actually, within a cyst, you can see the organism reproducing, and they stay like that, and then the cells come out once the good environment returns. The resting cysts are those typical dormant structure. There is no reproduction there. They just stay like that, and then they just come out alive when the good environment returns as well. This tree infers that the ancestral state is equally likely to be either resting cyst or no cyst, and these red circles, which are the reproductive cysts, they are really the derived state. So that's an ability that was not ancestral, that was gained. And what was cool, or what seemed cool, is that so these arrows represent which of the species are parasitic, and pretty much you see that any species that is capable of producing reproductive cyst also tends to be parasitic. Of course, this is not a quantitative analysis of this correlation between ability to produce reproductive cyst and ability to be parasitic. There is actually a way to quantify this correlation or test for this correlation, and we are also working on that once we have the maximum likelihood and Bayesian approach worked out here. But it's still a pretty cool pattern to kind of keep us going forward. And so I want to summarize this part by just kind of highlighting the effectiveness of ancestral character reconstruction to tell us a lot about evolution of a particular trait or evolutionary transition between different alternate states, also co-evolution of traits. Of course, there are caveats to this approach, a lot of caveats, specifically with ciliates. Not all branches of the phylogeny are well resolved, and so that is something that we are working on right now, how to deal with zero-length branches, because ancestral character state reconstruction really requires the branches to be at least well supported. And we also need to keep in mind that there could be a lot of missing data across the phylogeny. And so the analysis could be sensitive to how much missing data we have. But overall, at least it gives us some idea about how traits are evolving. And so this kind of analysis always ends up being worth doing to kind of get an idea of how, these species are living life in different environments. All right, I think I went over a little, but I just want to take one last minute to say thank you to a lot of people. Dr. Betsy Reed and Dr. Matt Escobar have been just really phenomenal here at Cal State San Marcos, kind of supporting my efforts in research. Dr. Reed is working on haptophytes, and she has worked on haptophytes for a long, long time. Like I said, Asher and Ihotu, I lucked out. This is phenomenal students and doing all the work. CSUSM also has been nice, supporting some assigned time or some research internal grants. And of course, Becky Zufal, who has always been awesome working with Ciliate. So thank you all, and I will be happy to take any questions if you may have.
Thank you so much. That was great.
Thank you. So
I appreciate it. We do have questions, for sure.
okay.
I want to start with mine, though.
Uh-huh.
I usually start with the students, but did you say that there's random chromosome loss? Like, could you imagine a situation when a mammal where we just we lost a chromosome? That would be death, right? There's
Yeah.
no survive this. So you're saying these things can literally lose, is it a certain kind of chromosome? And that's a huge amount of DNA. What's going on there?
Right. So you're talking about this, right? The parasexual loss. So It depends on which species. I don't think there are patterns, and honestly, it hasn't been studied a whole lot. What we do know is because of these chromosomal losses, you get a lot of diversity um, produced because of these processes. At the end, you get a lot of, and this is kind of like, to me, as an evolutionary biologist, it seems like the species is hedging bets. Depending upon what chromosome you have, you get one type of phenotype. If you have another chromosomal makeup, you have another type of phenotype, and so you're kind of hedging your bets, increasing the diversity, and then maybe one particular will survive. We honestly don't really know the evolutionary consequences of this, but this
For the
parasite,
developmental consequences. I mean, for me, it's like
right,
you can't just, with the development of complex multicellular organism, you
right,
can't just drop a chromosome. Uh,
right,
but maybe single cell eukaryotes, there are a bunch of genes that you can or cannot have because your development is simple, or
right.
is that what people are thinking?
Sure. I mean, I'm pretty sure many of these also end up not surviving. So we really get to see only the ones that survived. So there is a lot of way to, explore, well, what you need in order to survive and what you don't. But we do know that many species do show this, random loss of chromosomes and parasexual cycle. And it's just interesting and it's mind-boggling, like you said. It would be unthinkable in a multicellular species or almost.
Unthinkable. Yeah,
Yeah.
that's what I love about the microbial world is that you can, with bacteria, you can just throw in some genes like they're Legos, and they
Right.
work. So eukaryote, it's nice to see. It's interesting to see that eukaryotes, single cell eukaryotes, can have such a complex,
Yeah.
lifestyle with because they're so simple in some ways.
Right. So one thing I didn't mention was also in ciliates, and that's the other cool thing. So I talked about the somatic nucleus, and when the cell is dividing, this somatic nucleus actually doesn't undergo mitosis. The germline nucleus undergoes mitosis, so that's the canonical nucleus. But the somatic nucleus is it's what we call amitosis, and it's like a bag of chromosomes and it just pinches off and one cell gets a little bit, the other cell gets a little bit. There is no rhyme or reason how really exactly that they exactly get the same chromosomal makeup, and so that also generates a lot of phenotypic diversity. And so this amitosis process is also being studied quite a bit. It's like how do you just pinch off a nucleus and just survive? But
Great. Okay, so I'll ask some
what
the students. So, can you
sure.
talk about the interactions between these? I'm sure there's many interactions between the microbial eukaryotes and bacteria, for instance. But can you talk about some of those interactions?
Yeah, so I mean, most species eat bacteria, so the really interaction that they have is prey-predator. Ciliates, for example, they're mostly bacteriovorous. When they're asexually reproducing, they really just, feed on bacteria. So when you're growing them in the lab, there are very few that can grow in, just very synthetic media, but otherwise you provide them with bacterial culture. In natural environments, that's one of their ecological significances. They are an important part of food chain, and they will feed anything that you give. I don't think we know whether they need specific kind of or specific species of bacteria. Most of them seem generalists. They'll eat anything pretty much. But honestly, I don't think there is enough research along those lines either. So we wouldn't know even if they were specific.
Maybe they take some of their genes in, who knows?
Yeah,
Um,
it's
yeah,
possible.
take their genes in, use them eventually. Um, so someone had a question about the programs you use to create the phylogenetic trees and why you use them. I just want to talk to my students about we've learned about BLAST. Everyone's
Right.
using BLAST. We learned about phylogenetics, and we actually learned about, my students might not know this, but we
Mhm.
use parsimony to reconstruct ancestral nodes, which is
Yep.
essentially the same thing as we did it for amino acids, but you can do the
Ah, okay.
same thing for character states, right?
Right,
For
right.
parsimony. So we actually did that. But did you use a specific program for ancestral character states?
Well, we started out using Mesquite, but we are also right now trying out Phytol. So the issue that we are having with Mesquite, I think it considers, well, in Mesquite, we are not able to do maximum likelihood because of the zero length branches, and we are kind of restricted to maximum parsimony. So we are trying to move towards phi tools for that. Mesquite also, I think, if I remember correctly, it considers the phylogenetic tree to be completely reliable. And we definitely have some polytomies in our phylogenetic tree because ciliate phylogenies, there are some parts that are just not as resolved. You can kind of see that there are some nodes that are just not well supported. And so as far as building phylogenies is concerned, we are using RACSML, but otherwise, so the phylogeny is maximum likelihood, but when we do the ancestor characteristic reconstruction, it is maximum parsimony based. And currently in Mesquite, but I'm hoping that once I figure out a little nicks in phi tools, I'll be able to get the maximum likelihood and also Bayesian
But you're
approach working,
struggling with bioinformatics programs? I don't understand.
right?
That's like all we do is struggle with formats or like getting some software to work,
Yeah, like this
Oh,
format of matrix works with one, but not the other. And then especially in this case, we don't always have binary states for the characters. So a single species is not just cis versus no cis, it's like different kind of cis. So we also have to determine whether we put those like enforce binary nature of the characters versus, include different levels. So those are some of the nicks that we're trying to figure out.
we had some conjugal questions that are kind of similar
Mhm.
about the advantages of different, being, so for instance, we had a question about what might be advantages between alternating haploid and diploid phases. Another might be like, how does reproductive mode, sexual, parisexual, sexual, give an evolutionary advantage? And there's another one about germline versus somatic nuclei. Do these things, do people know how and why these various things contribute? Is there a select advantage? Is it like a random thing that just happened to work and they keep doing it? Um,
Unfortunately, I don't think we know about the adaptive significance. I mean, adaptive significance is hypothesized. For example, we can make pretty good arguments about why dormancy evolved, right? You can stay alive forever or you can survive forever. And that seems like a pretty good strategy. Cannibalism, we can also say a little bit about parasitic forms as well. But when it comes to haploid versus diploid, so when you look at the ploidy, people have shown there are advantages to being diploid. And I think this question has been asked in general, not just specifically in terms of microbial eukaryotes. It's like, why are many species diploid, right? And so as far as advantage of haploid versus diploid, we don't really know in these species, but it seems like, especially in some of the haptophytes, the haploid and diploid stages, they occupy distinct niches. So one exists in one kind of environment, the other exists in another kind of environment. And the simplest answer might be because it doesn't harm, right? And so it's kind of neutral. But unfortunately, I have the answer. It's like we just don't know. There are hypotheses that could be tested, but we just don't know whether this is better than just living as a haploid or than just living as a diploid.
Yeah, it's really interesting because in the haploid state, of course, you're exposing yourself much more because there's no way to cover up a bad gene.
Right.
And so it could be, I don't know if that happens, but if you have a bad gene or something doesn't work, that thing dies.
Yeah.
So, it doesn't continue in that sense, but for that organism, it's bad. For the
Right.
if you have a lot of offspring, maybe it's some of them will live, some of them will die, and then in the
Right.
next generation, you've kind of weeded out the bad genes.
Right.
Have anyone ever hypothesized that idea?
I I mean, there are hypotheses like that. I just don't think they have been tested empirically using these species. I mean, one could make an argument that because the haploid stage is non-motile, they can go anywhere in search for food. Whereas if you are diploid and you're, kind of staying in one place, if food comes to you, then that's great. But otherwise, you're not going to survive if you don't get to eat. So the arguments, I think there are both arguments for specific advantages and disadvantages of a particular stage, whether it's better to be haplodiplontic like this or just one kind of phase. It's anybody's guess at this point because we simply don't have data in these species.
Sometimes I like to remind people that evolution is like, well, it's not necessarily better, but it works.
Right.
It
Yeah.
works, it keeps on working. And until
Right.
it stops working, it's going to be around. It's not like they get a choice every time, like, well, I'd love to be diploid today, but I'm
Yeah.
really so no, you became haploid at some point. And maybe
Right.
you just can't, it's not like you can just flick a switch and reverse it, you know.
No, I mean, going from haploid to diploid, you need to mate with some other cell. And if there is no other cell to mate with, then that's it. Right. So, yeah, like exactly like you said, sometimes things evolve and are maintained evolutionary just because they are not harmful. They don't necessarily have to be better. It's just that they're not, it doesn't matter. So,
Yeah, that's good.
yeah.
Someone asked about the evolutionary speed at which microbial eukaryotes evolve. Can you compare? Are they really comparable, or is it, is it more like would you say it's in between, multicellular and bacteria? I mean, is there any way to look at that, or is it all just whatever?
Um, I think we see some patterns. For example, microbial eukaryotes that are obligatory parasitic or pathogenic, they show the similar pattern of, really rapid shifts and rapid evolution. Ciliates, for example, they have really rapidly diversified. But overall, I would say maybe they're somewhere between bacteria and metazoans. Bacteria are definitely super fast, I would say. Uh, Also, it has to, so for example, many microbial eukaryotes are just really slow growing. Their life cycles kind of take a lot longer than bacterial life cycles, for example. And I'm sure that directly matters into how fast they would evolve. So,
Some bacteria are very, very, very slow. They literally evolve
yeah,
for millions of years, but those are
yeah,
like stuck in the deep in the earth, and they only eat uranium. So,
yeah.
even the microbial world. But as far as I understand it, there's probably not as much horizontal gene transfer in the microbial eukaryotes as with like bacteria. Is that true?
I think that is fair to say. It's not non-existent. Fungi, which are also, some people do include at least some species of fungi under microbial eukaryotes. They have shown some HGT. But yeah, I would say bacteria probably do it better or more than microbial eukaryotes. That's not to say that they wouldn't have. I mean, for example, when studying haptophytes, I recently learned that even these coccolith-forming things, they have their own viruses, and the viruses infect them. And actually, one of the largest marine viruses known is the one that infects these coccoliths. I just recently learned that. And so I'm sure that these viruses and any other bacteria that infect them, they can, move genes around. But again, I'm not sure we have really looked. I definitely haven't looked into what are the rates of horizontal gene transfer in these species. Yeah.
Oh, that's cool.
Yeah.
We had a really interesting question here about potentially the idea of their differential cell environments, like how they differentiate. Is that way to, could you use that for maybe for more efficient means of manipulating human cell differentiation? Are there similar processes and patterns? This is a Frankensteinian question.
Yeah, I mean, I don't know if we can use these to manipulate human cells. I wouldn't go that far. I'm sure in theory it's possible, but one of the cool things that we have seen. So I have worked over the years. I worked on fungi. I'm working on these haptophytes right now. I worked on ciliates. I've kind of dabbled into many different groups, and often it seems like the pattern is that if you starve them, especially of nitrates, that's it. That's a trigger, and so there are some common triggers across the large evolutionary distances. So these are very large evolutionary distances across these groups. And pretty much in every single group, it's usually starvation that triggers something like sexual reproduction or this differentiation. So I would think there are common themes here. Whether you can use them to manipulate human cells, I would not be qualified to answer that question for sure. I have never really worked with human cells, so yeah.
You used reciprocal blast analysis.
Yes.
So this is a concept that my students, like I try to explain it once in a while, but how would you describe the theory behind what is reciprocal blast analysis, and how would you describe why it's useful?
Great question. So, reciprocal blast basically relies on this sort of hypothesis that when we obtain blast results, um, usually what we are looking for, our blast really finds, similar sequences based on sequence similarity. And it's really sequence similarity. It's not necessarily homology. So I always say that a lot of students think that similarity is basically the same thing as homology, but no, similarity is really just one aspect of homology. Homology is sequence similarity because of common ancestry. And so when BLAST finds similar sequences, we are assuming that they're similar because they have evolved from the common ancestor, and that's why they're similar, right? So if you use one sequence and as a query and found another sequence that is similar, if they're really homologous, meaning if they're really similar because of their common ancestry, you should be able to use that sequence that you found in reciprocal BLAST and hit that query sequence as the most similar, as you call it, or the most similar hit. And so that reciprocal BLAST kind of addresses this aspect of homology a little more than just doing one directional blast. I don't know if that
It
makes
could be
sense, but
part of a gene family,
right, it's
right?
part of a gene family.
So
It's the descendant
yeah,
that,
exactly.
yeah,
I always say that there is some distant homology, but the question is it direct descendant?
right,
That's really
right.
reciprocal blast. It seems very improbable that if you blast it one way and get that at top hit, and then you blast it back and still get this in the
Still
top
get
hit, then
exactly,
it's like improbable that that would happen by chance. That's
exactly.
kind of
Yeah,
how I've
that's
seen
a good way to put it. Yeah, yeah,
it. Yeah, it's like very
exactly.
unlikely if they're really not homologous, you know.
Yep,
Um,
yep.
yeah, no, this is great. I mean, I think we should probably close it off now. It's already 10
Oh
minutes
yeah,
after. I did have a question about the ancestral sequence reconstruction, but
sure.
I was just like, if you're missing a lot of lineage, can you simulate missing lineages and like
Yeah,
ask if you
that's
really get that and maybe even simulate because there's two options. One is that there's 27 independent evolutionary events, or it
yeah,
could be a whole bunch of losses. Things
right.
like that. Do you simulate the people try to simulate those things to see what
That's
like how accurate your stuff is?
Yeah, that could be the next stage. We haven't reached that yet, but that's a great idea to kind of see, especially in cases where you have a lot of missing data and that's influencing your whole distribution and your inference. So yeah.
At your zero, like these really short branches, I noticed can seriously, depending on the
Yeah,
method, it can really weight things, especially parsimony, because you're
yeah,
like overinflating very, very close, lineages, like
yeah.
a bunch of strains or something of
Right,
bacteria or whatever. Like, yeah,
right.
they'll have so they're going to overweight in the, thing. So I don't know. It's just an idea.
Yeah, no, for sure. That's why I call this work in progress. This is like the first pass analysis while we try to get working with those nicks. But yeah, we definitely need to move beyond parsimony and get some of those approaches worked out and see if we get the same result or what else changes
Oh,
for
I
sure.
love parsimony. Don't get me wrong. I love it. But I agree that, like even just simulations where you drop things out, put things back in, try stuff
Right.
in
Yeah,
any method. But I do think other methods, especially with the branch links, are
definitely.
much,
Yeah. Yeah, that's one of the biggest problems, the branch lines. Like
yeah,
it would be great if we have result phylogeny, but we don't. So yeah,
very cool. Well, it is really a pleasure to have you. This is such an
thank you.
interesting topic, and I hope people spend more money on microbial eukaryotes because they're fantastic, and they like produce like half of the oxygen in the ocean or something like that.
Yeah, yeah, they do some
Like,
very cool stuff. We ignore them way too much, I think, too. But
oh
yeah,
yeah,
I agree with you.
like every fourth breath or something, you breathe in oxygen made from a microbial eukaryote. There's
That's a
something
good way to
like
put it.
yeah, I mean like what I say is like you take one breath in, that's plants. You take another breath in, that's microbes. And even the plants are microbes, frankly, but that's a different story. But because of the chloroplast, right? But half of the oxygen comes from the ocean, and then a
Yeah,
huge
yeah,
percentage of that is from microbial eukaryotes, and we just like, they're
yeah.
just little things in the ocean.
That we know nothing about. Yeah, I
Oh,
so
no.
I when I teach microbiology, I tend to include them in the course syllabus somehow to make sure that they are represented there for sure. And I think
Ah,
if we
they're
keep doing
massive.
that,
They're
yeah, yeah,
massive players in global, uh, in the global climate.
yeah,
Absolutely mass it, and they just like, oh, we can't see them, so they don't exist. Let's go
Right.
study. Whales are
Yeah.
cool though. I'm not going to be against whales, but
Sure.
a lot more biomass. There's a lot more microbial.
There is definitely a lot more. And I think they are showing up more and more in microbiome studies too, because earlier it was just bacteria. And now we realize that, oh, well, but there is a large part that we just don't describe it if we don't look at microbial eukaryotes. So yeah, we'll know
Okay,
more.
great time, and thanks for coming to talk to us. That was really fun.
Thank you. I appreciate it.
And
Thank you
take
very
care.
much. Yep, you too. Bye.